Welcome back to the Deep Dive. Ready to go deeper into the world of biomolecules. Today we're tackling carbohydrates, going way beyond the basics, like what you'd find in an intro bio class.
Definitely. We're going to dive into some excerpts from a pretty advanced biochem textbook.
Yeah, perfect for all you advanced bio students out there. And trust me, even if you think you know your sugars, you might be surprised by what we uncover about saccharides and glycans.
It's time to think beyond just simple sugars, for sure.
Exactly. Okay, so maybe we should start by defining some terms. The textbook makes a distinction between carbohydrates and saccharides. Can you clarify that?
Yeah, so a lot of people use those terms interchangeably, carbohydrate and saccharide, but there's a subtle difference, you see.
Oh, okay.
Carbohydrates, strictly speaking, fit that CH2ON formula. Saccharides, though, that includes carbohydrates and all their modified forms, so it's a broader category.
So all carbs are saccharides, but not all saccharides are carbs.
Exactly. Think of saccharides as the bigger umbrella term.
Got it. And the textbook also throws around the term glycans. Is that the same as saccharides?
Yeah, pretty much. Glycans is, it's more for when you're talking about those chains of sugars like the oligosaccharides and polysaccharides.
Okay, but before we get too deep into, the chemical nitty-gritty, why are these molecules really important? Why should we care about all this?
Well, carbohydrates are essential. They're the core of the energy cycle.
Right. Plants use photosynthesis to capture energy from sunlight and store it in carbohydrates like starch.
And then we eat the plants or animals that have eaten plants, and we break down those carbs through respiration to power our cells. It's this beautiful cycle.
I see. But the textbook emphasizes that carbs aren't just about energy, right?
Oh, absolutely. They're incredibly versatile. Think about the structure of plant cell walls. Those are carbs.
The cellulose.
Exactly. Or even the tough exoskeletons of insects. That's chitin, another carb. They're even involved in complex processes like cell signaling and immune recognition.
Wow. So to really understand all that, we got to break down these complex molecules into their building blocks. So the textbook starts with the simplest sugars, the monosaccharides.
Right, the monosaccharides, those are like the individual letters of the carbohydrate alphabet. They're the foundation for everything else. Glucose, fructose, those are the familiar ones.
And the simplest of those are the trioses, of 3 carbons, right?
Yeah, you've got glyceraldehyde and dihydroxyacetone. And those two actually perfectly illustrate the 2 main classes of monosaccharides, aldoses and ketoses.
Okay, so glyceraldehyde with its aldehyde group is an aldose. And dihydroxyacetone, with its ketone group, represents the ketoses.
Precisely. See, even at this basic level, we start seeing variations in structure.
And I remember from organic chemistry that those variations can have big effects on how the molecules behave.
Oh, absolutely. And then there's chirality. Super important concept with sugars.
Right. Chirality, meaning the molecule can exist in 2 forms that are mirror images of each other, but not superimposable.
Yeah, like your left and right hand. So you've got diglyceraldehyde and L-glyceraldehyde. It's enantiomer.
And like with amino acids, the textbook points out that nature seems to prefer one enantiomer over the other.
Definitely. Demonosaccharides are way more common in biological systems.
Just like L-amino acids are the building blocks for proteins. Interesting how nature has those preferences. Okay, so that's trioses. What happens when we add more carbons to the sugar molecule? Like with tetroses, the 4-carbon sugars.
Things get, more complicated. That's when you start running into diastereomers. That's where they have the same chemical formula, but the atoms are arranged differently in space.
Not mirror images like enantiomers.
Right, not mirror images. And with every carbon you add, the number of possible isomers just explodes. It's like, whoa, suddenly you have this massive variety of carbohydrate structures.
Leading to a huge diversity of functions, I imagine.
Oh, absolutely. And then things get even more interesting with the bigger sugars. The textbook mentions that pentoses and hexases, those are the 5 and 6 carbon sugars, they prefer to form ring structures.
Because rings are more stable.
Exactly. It's all about stability. That ring forms when the carbonyl group reacts internally with a hydroxyl group.
So for a pentose like D-raves, you can get either a 5-membered ring or a 6-membered ring. And those are called furanose and pyranose, right?
Yep. Named after fern and pyrin, those cyclic compounds.
Clever. But the textbook also mentions that forming that ring creates a new chiral center, the anomeric carbon.
Right. So when the ring forms, the carbonyl carbon goes from being flat to tetrahedral. Now it can exist in 2 configurations, alpha or beta, and those are called anomers.
adding another layer of complexity.
It gets even more complex because these ring structures aren't static. They're constantly shifting between open chain forms and alpha and beta anomers.
And that's called mutarotation, right?
Yes, it's a dynamic equilibrium.
Okay, this is getting pretty intricate. I'm glad the textbook introduces Haworth's projections to help visualize all these cyclic sugar structures.
Haworth's projections are useful, but they are simplified, like a 2D snapshot. They don't really show the true 3D shape of the molecule.
So the rings aren't actually flat.
Nope. 5- and 6-membered rings pucker in 3 dimensions. These different puckered shapes are called conformational isomers, and they can have a big impact on how the sugar interacts with other molecules.
Okay, so those rings can pucker into all these different shapes, and the textbook specifically highlights a few key hexases: glucose, fructose, mannose, and galactose. Why those?
Well, they're the most common ones for sure, and they each have their own preferred ring form and play important roles.
Glucose, of course, that's the star player for energy.
Yeah, and fructose, that's the one that gives fruits and honey their sweetness.
Yeah.
Mannose, that's involved in attaching sugars to proteins, glycosylation.
And galactose is part of lactose, the sugar in milk.
Exactly. And it's also a component of a lot of glycolipids and glycoproteins. Those are, lipids and proteins with sugars attached.
Right, right. The textbook also points out that glucose and mannose are epimers, as are glucose and galactose.
Ah, yes, epimers. That's a specific kind of diastereomer.
Meaning they differ at just one chiral center.
Exactly. Glucose and mannose, they only differ at carbon 2. And glucose and galactose, they differ at carbon 4. But those tiny differences are super important.
Because enzymes are so specific.
Yeah. Enzymes can tell the difference between epimers. Often, an enzyme will only work with one specific epimer, not the other.
Amazing how these subtle differences matter so much. Okay, back to those ring conformations. The textbook dives into those chair and boat forms for pyranose rings.
Right, and usually the chair form is the more stable one. It's all about minimizing those steric clashes between the atoms.
So the chair form is kind of like a comfy armchair for the molecule.
Exactly. The bond angles in the chair conformation are closer to ideal, so there's less strain.
Makes sense. Now, the textbook also mentions sugars with more than 6 carbons, but those seem to be less common.
True, but there are some important ones, like cetoheptulose. It's a 7-carbon sugar that plays a key role in photosynthesis and the pentose phosphate pathway. Shows you that even those less common sugars can be pretty important.
Okay, so we've covered a lot of ground with monosaccharides. Now let's move on to those modified sugars, the ones with those extra groups attached.
Ah, yes. The sugar derivatives. They add a whole new level of complexity and functionality.
One of the key modifications the textbook talks about is phosphorylation. That's when a phosphate group gets added to the sugar.
Yeah, and that's often how sugars get activated for metabolic reactions. Think about ATP, adenosine triphosphate. That's a phosphorylated sugar.
Right. And those phosphorylated sugars are pretty acidic, right?
Yep, that phosphate group with all those hydroxyl groups, it can easily donate protons, and that acidity is often important for how they interact with enzymes.
Okay, so that's phosphorylation. The textbook also discusses sugar acids and lactones. How do those form?
Those are formed by oxidizing the sugar. So if you oxidize an aldose, you can get an aldonic acid, like gluconic acid, and then that acid can cyclize to form a lactone.
And what about uronic acids? The textbook mentions those as being important components of certain polysaccharides.
Ah, yes. Uronic acids have that carboxyl group at one end, which gives them unique properties. Like glucuronic acid is a key part of hyaluronic acid, that stuff that helps lubricate our joints.
So oxidation can lead to all sorts of interesting sugar derivatives. What about reduction?
Reduction of the sugar, that's when you reduce the carbonyl group. And that gives you alditols, which are also known as sugar alcohols.
Like sorbitol.
Yep, sorbitol is a good example. It's often used as a sweetener, but too much of it can cause problems like cataracts in people with diabetes.
Right, I remember reading about that. So even these seemingly small changes to the sugar molecule can have big impacts on health. Okay, and then there are the amino sugars. Those seem pretty important.
Oh yeah, amino sugars are everywhere. They're key components of lots of complex carbohydrates. Think about glucosamine and galactosamine. Those are two of the big ones.
And they often have modifications like N-acetylation, right?
Exactly. That's when you add an acetyl group to the amino group. And those modifications, they just add another layer of complexity, expanding what those sugars can do.
Amazing how much diversity we see, even with these modified monosaccharides. Now, the textbook shifts gears and starts talking about glycosides.
Glycosides, those are formed when a sugar reacts with another molecule, usually through a condensation reaction, forming a glycosidic bond.
So that bond links the anomeric carbon of the sugar to like an oxygen, nitrogen, or sulfur atom on the other molecule.
Exactly. And that other molecule, it could be another sugar, a lipid, a protein, or even something small like methanol.
And those glycosidic bonds, they could be involved in some pretty crazy stuff, right? The textbook mentions some toxic glycosides.
Yeah, like uabane, that's a cardiac glycoside. It messes with the sodium-potassium pump in heart cells.
And amygdalin, that releases cyanide when it's broken down.
Exactly. Shows you how adding that glycosidic bond can completely change a molecule's properties. Makes you appreciate the importance of understanding glycosidic bonds in, like, pharmacology and toxicology.
For sure. Okay, so we've talked about individual sugar units, but now let's move on to those bigger chains, the oligosaccharides.
Oligosaccharides, those are like the stepping stones between monosaccharides and the really big polysaccharides. They're chains of a few sugar units linked together by those glycosidic bonds.
And the simplest examples are the disaccharides, right? Just two sugar units linked together.
Yep, disaccharides, they're super important, playing roles in like energy storage and as building blocks for bigger carbs.
The textbook mentions sucrose, lactose, and trehalose as examples. So what makes those different from each other?
Well, a few things. First, the actual sugars involved, like sucrose is glucose and fructose, lactose is galactose and glucose, and trehalose is 2 glucose units.
Okay, so that's one difference. What else?
The carbons involved in the linkage. Like, is it a 1-4 linkage or a 1-2 linkage, And then the order of the sugars matters, too, if they're different.
So even with just 2 sugars, there's a lot of variation.
Oh, yeah, tons. And don't forget about the alpha and beta configurations of that anomeric hydroxyl group. That can make a big difference, too.
Right, that determines whether the linkage is alpha or beta. Okay, so we've got all these different disaccharides, but how are they made?
Well, forming that glycosidic bond, it's a condensation reaction, so water gets released.
But that reaction by itself isn't energetically favorable.
Right, exactly. So cells use activated sugar molecules, usually nucleotide-linked sugars, to drive the reaction.
Like UDP galactose, which is used to make lactose.
Bingo. That UDP acts as an energy carrier, kind of like ATP. So when the UDP galactose is hydrolyzed, that releases energy, making the whole process go forward.
Clever. The cell uses that energy to overcome the energetic barrier of forming the glycosidic bond.
Yep, and there are specific enzymes that catalyze these reactions, the glycosyl transferases.
So lactose synthase, the enzyme that makes lactose, that's a glycosyl transferase.
Precisely. It transfers the galactose from UDP galactose to glucose, forming that specific 1M linkage. And each glycosyl transferase is really picky about which sugar it transfers, which molecule it transfers it to, and what kind of linkage it forms.
It's like a whole assembly line with these specific enzymes putting together these complex carbohydrates.
Yeah.
Amazing. Okay, so we've gone from single sugars to pairs of sugars. Now let's tackle those giant polymers, the polysaccharides.
Ah, the polysaccharides. They're like the skyscrapers of the sugar world.
Yeah.
So much bigger and more complex than those little monosaccharides and disaccharides.
Now, the textbook points out that polysaccharides are different from, like, proteins and nucleic acids in a few key ways.
Definitely. Proteins and nucleic acids, those usually have a defined sequence and length. Polysaccharides, they can be all over the place.
Meaning they can be linear or branched, and they can be made up of the same sugar unit over and over, or they can have different sugar units mixed.
Yeah, exactly. And that gives them amazing flexibility. They can be used for all sorts of things, like storing energy or providing structural support.
Okay, let's dig into those functions. The textbook starts with storage polysaccharides, which makes sense. Those are like the cell's pantry. So yeah, those storage polysaccharides, they're like the cell's backup energy supply, ready to be broken down when needed. And the textbook focuses on starch in plants and glycogen in animals as the main examples of those storage molecules.
Right, exactly. Starch, it's found in those little granules inside plant cells, and it's actually a mix of 2 different polymers of glucose. Amylose and amylopectin.
And both of those are just long chains glucose linked together.
Well, amylose is a street chain, all those glucose units connected by those alpha 1-4 glycosidic bonds. But amylopectin, it's branched. It has those alpha 1-4 linkages, but then it also has these alpha 1-6 linkages that create the branches.
So it's like amylopectin has a main chain of glucose, and then every so often there's a branch point where another chain of glucose sticks out.
Exactly. And those branch points, they're super important because they create all these non-reducing ends where enzymes can come in and chop off glucose units. So when the plant needs energy, it can quickly mobilize glucose from those branch points.
That makes sense. And glycogen, that's the main storage polysaccharide in animals, right? Is that similar in structure to amylopectin?
Yeah, it's very similar, but even more highly branched than amylopectin. Lots and lots of branch points.
Makes sense for animals, right?
Yeah.
We tend to need energy quickly, so having all those branch points allows for rapid glucose release.
And the textbook mentions that these polysaccharides also have these specific 3D shapes, right? Like they're not just these floppy chains.
Oh yeah, the shape is definitely important. Amylose, for example, since it's a straight chain, it tends to form this helix in solution.
So it coils up on itself.
Exactly. It's like a tightly wound spiral staircase, and that allows for really efficient packing of those glucose units.
So amylose is compact, perfect for long-term storage. But amylopectin and glycogen, with all those branches, they probably have a different shape. Right, they're more globular, kind of like a tangled ball of yarn. That shape, with all those exposed ends, it makes it easier for enzymes to access and break down the glycogen when the cell needs quick energy.
Fascinating how those structural differences match the different needs of plants and animals. Okay, so we've talked about energy storage. Now let's switch gears and talk about those structural polysaccharides, the ones that give strength and support.
Ah, yes, the building blocks of the biological world. And in plants, the king of structural polysaccharides is cellulose. I mean, it's the most abundant biopolymer on the planet.
Yes, cellulose, it makes up those tough plant cell walls. And it's amazing to think that it's just glucose units linked together, just like starch.
Right, but there's a crucial difference, the type of linkage. Cellulose has those beta-1 for glycosidic bonds, while starch has alpha-1 for 4 linkages.
And that small difference, it makes a huge difference in how those chains pack together. Real.
Huge difference. Those beta-1 to 4 linkages force the glucose chains into this straight, extended conformation. They can pack really tightly together, forming these strong fibers.
Like those long, straight logs in a log cabin, all lined up perfectly.
Exactly. And all those hydrogen bonds between the chains, that adds even more strength. That's why plant cell walls are so tough.
But that also means we can't digest cellulose, right? We don't have the enzyme to break those beta-1 to 4 bonds.
Most animals don't. We lack cellulose, the enzyme that can break down cellulose. But some animals, like cows and termites, they've got these symbiotic bacteria in their guts that produce cellulose. That's how they can digest all that plant material.
Amazing how nature works that out. So cellulose is the star structural polysaccharide in plants. What about chitin? The textbook mentions that as another important one.
Yes, chitin. That's what gives those hard exoskeletons to insects, crustaceans, all those arthropods. And it's found in fungi, too.
So chitin, it's similar in structure to cellulose, but with a twist, right?
Right. It's still a polymer with beta-1 to 4 linkages, but the monomer is N-acetylglucosamine. It's like glucose, but with an amino group that's been acetylated.
And that little change makes a big difference.
It does. That N-acetyl group, it allows for even stronger hydrogen bonding between the chitin chains, making those exoskeletons super tough and rigid.
It's incredible how those subtle chemical modifications can have such dramatic effects on a material's properties.
Okay, and then the textbook jumps to glycosaminoglycans, another class of structural polysaccharides. But these are found in vertebrates.
Glycosaminoglycans, yeah, they're fascinating. They're key components of connective tissues, cartilage, all that good stuff.
Yeah.
And they're different from cellulose and chitin in a few ways.
They're heteropolysaccharides, meaning they're made up of different sugar units, often those modified ones like N-acetylglucosamine and N-acetylgalactosamine. And the textbook emphasizes that they have a lot of negative charge.
Yeah, that negative charge is super important. Those repeating disaccharide units often have sulfate groups and carboxyl groups hanging off of them, and those are negatively charged.
Oh.
That attracts water molecules, creating that gel-like consistency that you see in connective tissues and cartilage.
So it's their ability to bind water that makes them so important for cushioning and lubrication. Exactly. And the textbook gives this cool example of the proteoglycan complex in cartilage. It's like this intricate structure where these long hyaluronic acid molecules act as a backbone, and then you have core proteins attached, and then those proteins have chondroitin sulfate and keratin sulfate chains attached to them. It's like a giant branched molecule.
Wow, so complex. And all those negatively charged sulfate groups, they're attracting water, creating this gel-like matrix.
Exactly. And that matrix, it interacts with collagen, which is another structural protein, and together they provide the strength and resilience of cartilage.
It's amazing how these molecules all work together to create these complex tissues. And glycosaminoglycans, they're not just for structure, right? The textbook mentions hyaluronic acid being important for lubrication in joints, and heparin, which is a natural anticoagulant.
Right, so much variety in their functions. It's really cool. Okay, and then the textbook takes us into the microbial world, talking about those polysaccharides and bacterial cell walls.
Yeah, those bacterial cell walls, they're so important for protecting those bacteria from harsh environments. And their composition is what makes the Gram stain work, right?
Exactly. The Gram stain, it's one of the most fundamental tools in microbiology. It lets us quickly tell the difference between gram-positive bacteria and gram-negative bacteria based on how much peptidoglycan they have in their cell walls.
So gram-positive bacteria have a thick layer of peptidoglycan, and they hold onto that crystal violet stain. Gram-negative bacteria, they have a thinner layer, and they don't stain as strongly.
Precisely. So what exactly is peptidoglycan? It's this amazing, unique polymer. It's got these polysaccharide chains made up of alternating units of N-acetylglucosamine, AGA, and N-acetylmuramic acid, NAM.
And each of those NA units, they've got a little peptide chain attached to them.
Yeah, a tetrapeptide chain. And get this, those tetrapeptides are cross-linked to each other, creating the strong mesh-like structure that surrounds the bacterial cell.
It's like a molecular chain link fence protecting the bacterium.
Perfect analogy. And the textbook points out something really cool about those tetrapeptides. They contain both D and L amino acids.
Which is unusual, right? Most proteins are only made of amino acids.
Exactly. It's a rare exception. And those cross-links, they're super important for peptidoglycan's strength. They help it resist osmotic pressure and keep the bacterial cell from bursting.
So it's this intricate network of sugar chains and peptide cross-links that gives bacterial cell walls their strength and rigidity. It's amazing.
It really is. We've covered so much ground today, from those simple monosaccharides to those massive, complex polysaccharides. And we've seen how they're involved in everything from energy storage to structural support to signaling to even protecting bacteria.
I feel like I have a whole new appreciation for carbohydrates. There's so much more than just sugars.
Absolutely. And I hope our listeners feel the same way. It's such a fascinating area of biochemistry, and there's so much more to discover.
Well said. And on that note, we'll wrap up this deep dive into the world of carbohydrates. Thanks for joining us. Until next time, keep exploring.